Delighted to be here. It's been a great 2 days so far. And so I'll be talking about Mendelian randomization. For those of you who don't know what this is, well, I'll be introducing it very soon. But just quick show of hands, who has heard of it yet? Okay. Who hasn't? Okay, good. So, right, so the motivation. So we are interested in estimating the effect of an exposure or treatment or an outcome of interest. So here an exposure or treatment is basically the variable we want, whose causal effect we want to estimate. So for instance, suppose we want to know the effect of height on income, or the causal effect of height on income. Well, as we know, causal inference with purely correlational data is usually problematic because there are confounders that may influence the relationship between the exposure and the outcome. I mean, all I'm saying here is that correlation is not equal causation, as we all know. So for instance, if you regress income on height, well, you cannot interpret the coefficient on height as a causal effect. And the gold standard to make causal inference is randomized controlled trials, or RCTs for short. Unfortunately, for many cases, it's not possible to implement RCTs. So for instance, if you want to see the causal effect of height on income, we cannot just randomize height across people and see its effect on income. It's just not feasible, nor ethically possible anyways. However, nature has provided a natural experiment because individual genotypes are assigned randomly at birth. So if individual genotypes directly influence the exposure, in this case height for the example, and only influence the outcome, income, via the exposure, then a research design similar to RCTs can be used. Okay? So that's the basic idea of Mendelian randomization, is to use this randomization of alleles at conception to try to tease out a causal effect of a treatment of interest or an exposure of interest. Okay? I'll be saying exposure throughout, but when I say exposure, it's also a treatment variable. It's also known as the risk factor, and there's different names for it, or intermediate variable. Okay, here's a diagram comparing randomized controlled trial to Mendelian randomization. So let's focus on the right-hand side to start with. A randomized controlled trial, you first have a randomization method, usually a computer pseudo-random number. And then some people are allocated into an exposed intervention, and the other group or the other people are assigned to the control group. And then, well, the exposed get the intervention, and then we can compare the outcome between the exposed and the control group. Now, the idea of Mendelian randomization is quite similar, except that instead of having a computer pseudo-random number, we have the random segregation of alleles at conception. So basically, think of the example again of height and income. And suppose we want to see a causal effect of height. And suppose there is one allele that really increases height, and the other allele does not. Okay, for the sake of the argument, we know from the past few days that it's not the case that there's just one allele that increases height. But for this example, suppose there is. Well, since some people will have this allele, and so they'll be exposed to the increased height treatment, and others will have the other allele, so they'll be a control group. And then we can compare the outcomes between those who have the tall allele and those who don't. Okay? We can compare the income. Now, the key difference with Mendelian randomization here is that in Mendelian randomization, we really know what happens to the people who are exposed because we can really implement the intervention ourselves. I'm sorry, this is in randomized controlled trials, in RCTs. We can really control the intervention ourselves. However, in Mendelian randomization, we don't control the intervention ourselves. We have to trust nature. We have to trust that nature implements this intervention itself. So we have to trust that nature, by allocating the tall allele, makes people taller and does not introduce any other confounders. And this latter issue is actually the key issue that we'll be spending some time discussing to make sure that there's no confounders when using Mendelian randomization. Okay, here's a roadmap for what we'll cover for the rest of this lecture. So first I'll be giving you a 3 or 4 slide crash course on instrumental variables and see how they relate to Mendelian randomization. Second, I'll talk about Mendelian randomization and the relevance and exogeneity conditions. These are the 2 conditions that are needed to have valid instruments and instrumental variables, and they also apply in Mendelian randomization. Then I'll talk about how to use control variables. I'll give some examples from the literature. And then I'll talk about MR-Egger regression, which is a new method that's been developed a few years ago that allows us to do Mendelian randomization under weaker assumptions. Okay? To start with, let's talk about instrumental variables. Here's the 3 or 4 slide crash course in instrumental variables. So again, show of hand, who is very familiar with instrumental variable? Who is not very familiar? A few people? Okay, good. So at least for those who are very familiar, please bear with me. This will be repetitive, redundant for you, but it's important to cover the basics. The basis first because, well, Mendelian randomization, simply put, is using genes as instrumental variables. So you need to have a solid understanding of this. So we wish to estimate the causal effect of the exposure, or treatment, E, on the outcome Y. Okay, so again, the effect of income on well-being or the effect of height on income. This has been our running example so far. If we estimate the simple linear specification of Y is equal to the exposure beta plus epsilon, well, as we know, we cannot necessarily estimate beta as a causal effect because there might be omitted variable bias. For instance, if the covariance between the exposure E and the error term epsilon is not equal to 0, then beta hat will be biased due to omitted variable bias. So this is from elementary regression analysis. Now just recall, these are the two equations we've seen so far. But suppose instead that there exist instruments X such that the exposure is equal to X times gamma plus Xi, okay? Well, and suppose that the following 2 conditions hold. First, suppose that gamma is not equal to 0. So that's called the relevance condition. And it states that instruments have non-zero effect on the exposure. So in other words, in this equation 2 here, the instruments X need to be related to the exposure, else they're irrelevant, okay? The second condition is called the exogeneity condition. And this basically says that X is orthogonal to epsilon. Okay, so basically X is typically called the instruments or the instrumental variables. And they have to be orthogonal to the epsilon from equation 1 here, okay? And that's called the exogeneity condition. And it says that conditional on the exposure, the instruments must be independent on the outcome, okay? So again, I'll recap. I'll go in more detail in this on the next slide, but still I want you to get this very clearly. The relevance condition is simply that gamma is not equal to 0. And the exogeneity condition is that the X in this equation here, in the second equation, is orthogonal, or is not related, to the epsilon in the first equation, okay? All right, let's put this graphically. So I'll be using these path diagrams a bit, and throughout, the convention is that, well, the circles are variables, and then the direct arrows indicate causality, and then the dotted arrow with 2-sided indicate correlations, okay? So here we see equation 1 says that Y is equal the exposure beta plus epsilon. So again, you have epsilon impacts Y, and E impacts Y directly also. And then the second equation can also be seen here. We have E is equal X gamma plus Xi, so E is caused by X and also Xi, okay? Now the relevance condition simply says that gamma is not equal to 0, okay? And the exogeneity condition states that there is no association or no correlation between X and epsilon, okay? So now the key between instrumental variable, I'll provide the intuition, then we'll do the formalities after. So the key intuition here is that X, okay, when X varies, epsilon doesn't vary. So it's possible to vary X, and this in turn causes variation in E that is independent of epsilon, okay, because X is independent of epsilon. So basically varying X causes variation in E that's orthogonal to epsilon. And then from this, we can try to tease out the causal effect of E on Y, okay? So that's the basic idea. So what we'll do formally is we substitute equation 2 into equation 1. And that's what it gives. Y is equal X gamma plus Xi beta plus epsilon. So here I've just substituted equation 2 for epsilon, for E here. And that's equal to X gamma beta plus a collection of the error terms, which is equal to E hat beta plus E double dot, okay? Where E double dot is just a new way of writing the collection of error terms. Okay? Now, the key insight for this is that if the exogeneity condition is satisfied, then E hat is equal to X gamma will be orthogonal to E double dot. Okay? And the reason for this is that E double dot is equal to psi beta plus epsilon, and then X is orthogonal to psi because that's from this equation here, okay, by construction. And then X is also orthogonal to epsilon because that's the exogeneity condition, right? So everyone sees that? What we've done essentially is we rewrote Y, okay, as we rewrote Y to be equal to E hat beta plus E double dot. But the key here is that in this equation here, E hat is orthogonal to E double dot, okay? So we can now estimate this equation and get an unbiased estimate of beta, okay? So that's the basic idea of instrumental variables, okay? Okay, that's just repeating what I said. Therefore, if we regress Y on E hat, which is equal to X gamma hat, then which is the predicted value of E, then we can obtain a consistent estimate of beta, okay? So is that clear for everyone? And, okay. So in terms of, okay, so here's going back over this in more detail just to make sure I really hammer this point home. So here I'm just rewriting the key equations. This is equation 1 and 2 again. Then we have E hat, which is the predicted value of E, okay? It's equal to X gamma, essentially, and that's orthogonal to E double dot. And we just estimate this equation here, okay? And, okay, I put the path diagram again here, but I think I already went in depth around in this. I won't repeat. In terms of terminology, we call X here the instrument for E, okay? X is the instrument for E hat. And the reason for this is because it allows us to instrument E in the sense that it allows us to create a variable E hat whose variation is orthogonal to epsilon, okay? And then this instrumented E hat is used in this equation, okay? So that's where. Okay, here's a famous, let me just pause. Any questions so far? Yeah?
So is the assumption, I mean, implicit in here, there's no direct link between X and Y, that the effect of X only goes through E. But is that assumption the same as the assumption of the exogeneity condition?
Oh yeah, you're right. I'll go in more depth into this. So here I was simplifying a bit. So what Matt is saying, if I understand well, is that there's no direct arrow from X to Y in this diagram. Actually, you're right that I could have, to be complete, I could have put a direct arrow from X to Y and an X over it to say we don't want that. But here, to simplify, I kind of assumed this was included into this. So actually, I'll go in more depth in a few slides on that. But yeah, to summarize, basically, the exogeneity condition rules out this type of interaction, but also rules out direct interaction between X and Y, which actually is also subsumed in this. But okay, here's a very famous example in the instrumental variable literature in economics. And this is a paper called Lifetime Earnings in the Vietnam Era Draft Lottery from Joshua Ingres and the ER 1990. And here, Y, the dependent variable, the outcome was lifetime earnings, so how much money do you earn in your life? Then the exposure variable was veteran status, and I'll describe this in a second. X was random draft lottery to be evaluated to enter the military during the Vietnam War period. Okay, so to give some context, there was a debate in the literature as to whether serving in the military had a causal negative effect on income. Observationally, there was a clear correlation, negative correlation between having served in the military and income after, like for people who have served and then go in the labor market. But the question was it a causal effect, or was it that people who tend to serve in the military self-select in such a way that they have other features that is also negatively correlated with income? Okay, so then during the Vietnam War, there was this draft lottery where basically people, when it was a certain birth year, turned to be enrolled in the military, there was a draft which picked several birth dates, let's say July 20 and May 17 and like whatever. Basically, they pick a bunch of birth dates, and everyone in that year born on these birth dates had to show up at a military induction center and then they had an exam, and then if they were healthy and functional and satisfied certain criteria, they were shipped to Vietnam. They were conscripted and typically shipped to Vietnam. So basically, we have a very good instrument here because the relevance condition here holds. Being selected in the lottery increased the probability of induction in the military. By definition, that's what the lottery did, essentially. It was to decide who was going to be conscripted versus not. And the exogeneity condition holds because the lottery was random, so independent of other variables, and so it was independent of other variables. And then common sense suggests that it had no direct effect on earnings, right? So based on this, Angress finds that long after their service in Vietnam was ended, the earnings of white veterans were approximately 15% less than the earnings of comparable non-veterans. So basically, he found a causal negative effect of serving in the military on earnings. Actually, in terms of to calibrate this, this is not really related to genetics, but this is equivalent to about 2 years of income, 2 years of experience in the labor market, and that's typically the amount of time people spent in the military. So this estimate makes perfect sense. It's like people were inducted in military, lost 2 years of labor market experience, and this translated in lower wages, essentially. Okay, now instrumental variable and Mendelian randomization. So Mendelian randomization is essentially just using genes as instruments. So for example, using Mendelian randomization to estimate the causal effect of height on income would be y would be income, the exposure would be height, and x would be genetic variants that directly influence height but have no effect on income except via height. Okay, and we'll spend quite a few slides going deeper into what I mean by this here when I say that they directly influence height but have no direct effect on income except via height. Okay? So that's the idea of Mendelian randomization. Okay, now let's look further into those two conditions, the relevance and the exogeneity conditions. So first, Mendelian randomization and the relevance condition. So recall the relevance conditions. So the genetic variance must have non-zero effects on the exposure, or gamma must be not equal to zero, must not be zero, basically. Okay, so that's the relevance condition. Well, in the context of Mendelian randomization, basically this, okay, I'll get to this in a second. So basically then, if gamma is equal to 0, then the exposure, or the predicted value of the exposure E hat, will be equal to X gamma hat, and will only capture noise, okay, of course. So it's important to use genetic variants X that are known to affect the exposure E, okay? To date, however, many Mendelian randomization studies have employed genetic variants whose effects have only been documented in candidate gene studies. And typically, these have very small samples and have a poor replication record, as we've already discussed in the past. So in other words, it's not even clear that in many of the Mendelian randomization studies that have been published that the relevance condition holds. It's very possible that they've used genetic markers that actually have no effect on the exposure, okay? Now, that's not the case for all studies, but quite a few of them, I suspect that's the case. Okay. Another important thing to keep in mind is it's not sufficient to have non-zero effects, okay? It's not sufficient to have non-zero effects. Because if X is a weak instrument for E, i.e. if the association between X and E is weak, okay, then the resulting IV estimates may be severely biased. And this is known as the problem of weak instruments, and there's the whole literature in econometrics about that, with the references here by Bond et al. And Steiger and Stock, Econometrica 97. But the rule of thumb that has typically is that to ensure that your instruments are not too weak, it's good that the F-test, that the F-stat from the F-test of the joint significance of the instruments in equation 2 should be at least 10. Okay, so remember equation 2 is basically just regressing the exposure is equal to gamma x plus xi. Well, if you do an F-test for the null hypothesis that gamma is equal to 0, then the F-stat should be greater than 10, okay? That's the rule of thumb.
So this weak instrument issue is more about the statistical significance of that association and is about the actual strength of the effect.
Correct, correct. In practice, that's, I mean, it's a rough rule of thumb, but yes, it gets at that, yeah. Right, okay, so basically we need to make sure that the genetic markers we use do influence the exposure. I mean, that makes sense, but Now, the more difficult condition to satisfy is the exogeneity condition. So recall again that this says that conditional on the exposure, the genetic variance must be independent on the outcome, or x must be orthogonal of epsilon to epsilon, or x and epsilon must be orthogonal. Well, one implication of this is that genetic variance must be independent of any confounding factors that affect both the exposure E and the outcome. Okay, so graphically, we have x that causes E, that causes Y, and suppose we have some confounder. Well, that can be a problem, so we don't want that, okay? Can anyone think of a situation where we would have such a confounder? Okay, think of something that has probably come up pretty often in the last week. With something, we want some variable that's correlated with the genetic variant and the outcome, not only via the exposure. Yes. Situation when this fails, population stratification. So suppose, here's a simple example, suppose genetic variant X vary between English and Scots, okay? So suppose that E is height and Scots are taller for non-genetic reasons. Suppose that Y is income and English incomes are higher for non-genetic reasons, okay? Well, then what we'll have is that there's some correlation between X and because genetic variants vary between English and Scots. So there's a correlation between X and being a Scot, and also a correlation between being a Scot and income. But this has nothing to do with genetics and nothing to do with what we want to look at, and this can bias the IV estimates. Okay. So population stratification can be a cause of bias. So that's why we need to control for the principal components. Hopefully that's sufficient, but who knows? There might be residual stratification, but okay, so we don't want that. Again, the animation. All right. All right. Another important thing for the exogeneity condition to hold is that the genetic variant can only affect Y via E. And that's called the exclusion restriction. That's what Matt was referring to earlier. Okay? Graphically, we have X causing the exposure and the exposure causing Y. And then we don't want, we could have a direct effect on top of this from X to Y. But we don't want that, okay? That would be violating the exclusion restriction, okay? So can someone think of a situation when this fails? Yep. Exactly, pleiotropy. Good, you guys are good. Or girls are good, actually. Suppose genetic, oh yeah, you have the slide. Doesn't matter. I'll still give points for people who get the right answer. All right. So suppose genetic variance X directly affect both height and, so to understand this, suppose the genetic variance X directly impact height, okay, so this is gamma here, and income, okay? So that means the impact income not only via height, but also directly, okay? So for instance, think of a genetic variant that directly affects height by affecting bone length, so that's gamma, and also affects income independently of height by affecting labor market skills. Okay, that's, how you call that, phi? Right. So, and then the variant may indirectly affect income via its effect on height. That would be beta, but that's basically not necessary. But in this case, the exclusion restriction or the exogeneity condition will fail, and we'll get a biased estimate. Okay, so now I know you have the slides, but still to encourage class participation, what's another situation when this fails? Someone? I won't move before. Anyone? LD? LD? Great. All right, so again, this is the same graph as before. We don't want this. Another situation when this fails when there's linkage disequilibrium. So suppose genetic variant X directly affect height, and/or in LD with variants that directly affect income. So it's basically the same reasoning as pleiotropy, except here the mechanism is LD. Okay? And again, the IV estimates will be biased for the same reason as before. Okay. Now, this slide I wrote not covered in the lecture because I don't expect I'll have enough time to cover everything. I'll still go through, I'll just mention very quickly what this is without going into detail. But basically, another way the exogeneity condition can fail, or via a failing exclusion restriction, is when parents directly influence the income, or the outcome, okay? And the reasoning here, I'll let you go through the example on your own, but the idea here is that you share your genes with your parents, so even if, your genes don't influence the outcome via your trait directly, it's possible that your parents will influence the outcome, and that could still cause failure of the exclusion restriction. Okay? Okay, so to recap, so here's a recap of everything we've seen so far, essentially. So the relevance condition states that genetic variance must have non-zero effect on the exposure. In other words, gamma is not equal to 0. So genetic variance with weak effects can also be problematic, and that's the problem of weak instruments, as we mentioned. Okay, also there's the exogeneity condition, which says that conditional on the exposure, the genetic variance must be independent of the outcome. In other words, x must be independent to epsilon. So just to make sure everyone understands this language, when I say conditional on the exposure, the genetic variance must be independent of the outcome, what I'm saying is we have y is equal to e beta plus epsilon. So conditional on exposure means y conditional e. So that's basically equal to epsilon, okay? So we're saying is conditional on the exposure, we're saying epsilon must be orthogonal to the genetic variant x, okay? Just so everyone understands the language. So that's what this says. Okay, so the genetic, okay, and to recap again, that means the genetic variance must be independent of any confounding factors that affect both the exposure E and the outcome Y. And this may fail when there's uncontrolled for population stratification. And the genetic variance can only affect Y via X, and that's the exclusion restriction, which may fail when there's pleiotropy, or LD. And it also can fail if the parents directly influence the income, as I mentioned, without going in depth into. For those who want a more in-depth treatment of this, a good paper is von Inke et al. In the Journal of Health Economics, 2016. Okay, good. Any questions so far? Okay, good. So now let's talk about control variables. So far, we haven't used any control variables. Well, when there are control variables, the relevance and exogeneity conditions still apply, but must all conditional on the control variables. So basically, we'll just restate the conditions, but adding conditional and control variables. So the relevance condition becomes that the genetic variance must have non-zero effects on the exposure, conditional on the control variables. And the exogeneity condition becomes that conditional on the exposure and on the control variables, the genetic variance must be independent of the outcome, okay? But that raises the question, when to include control variables in practice? Well, the exogeneity condition may fail when appropriate control variables are not included. So for instance, if there is population stratification and the top principal components of the genetic relatedness matrix are not included, then this exogeneity condition may fail, okay? So in addition, including control variables may increase the precision of the estimates, okay? That's, as always, in econometrics. If you have nuisance variable, it's good to control for them. However, in the specific case of Mendelian randomization and actually genetic studies in general, it's important not to control for downstream covariates that may be influenced by the exposure or outcome, okay? So for instance, including a dummy for urban versus rural residents may bias estimates of the effect of income of height on income if height or income affects preferences for living in cities, okay? So, yeah.
So when we were talking about just the general case last week about controlling for things that may be heritable, like a non-IV setting, something we were saying was that controlling for it is biased in the sense that it doesn't give you the total genetic effect. But if you just add the control and you want to say what's the effect of the treatment through channels other than this one that you're controlling for, do things still go through? Would that still be a right interpretation? Does that make sense? What I'm asking?
It made sense until the question, the last sentence. So I think, well.
So I'm just asking. So before we said if we controlled for something, so we have the SNP and outcome of interest.
Yep.
And there was maybe some pathway that the SNP goes through to get to the outcome of interest, right? So if we controlled for this thing in a standard non-IV setting, what we could say is that the coefficient that we get here is the effect of the SNP on the outcome, excluding this
conditional on this channel, yeah, or excluding.
Does the IV have the same interpretation
Yeah.
if you control for a downstream covariate?
Yeah, I'm pretty sure the answer is yes, yeah. So basically what Patrick is saying is, actually, if you want to, I'm saying here it's important not to control for downstream variables or mediating covariates, but except in the case where the effect of interest is one where you want to know the effect of, what was this example again? you want to know the effect of height on income conditioning on rural versus urban residents. Well, then in this case, you want to control for it, right? But in general, you want to be careful not to control for any mediating or other covariates like that because they could bias the effect of it. You have to know what you're doing, essentially, okay? So in general, it's advisable to control for the top PCs, okay, to control population stratification, as well as for sex and age, because these increase the precision of the estimates, and because these cannot be downstream variables because they're not correlated with the genetic variance. I mean, if you exclude the sex chromosome, which we usually do. So basically, for these reasons, we typically control only for these, okay? Okay, good. So now let's cover some examples from the literature. And I'll ask for your input. Now, it's not all in the slides, so you'll have to think on your feet and not just be able to read from there. Yeah?
Before we go on, I mean, the whole issue of what you control for. I mean,
Yep.
the example that you gave with the draft, it's kind of nice because there you really have something that will not affect anything else, and then it's a very clean issue. But here, even controlling for age may be something that you may be worried, right? Let's say that you are looking at earlier where perhaps height may affect if you actually survive to a certain age. So then age becomes something that you would not want to control according to what you're saying. But if I'm trying to think about the direct effect of height on my income, then I really want to control for everything, every other channel through which, which goes back to Patrick's point, I guess. Every other channel through which height may have affected income, but it's not the direct effect, right?
Yeah, no, it's a good point, actually. Whoops,
And
what have I done?
And then given that we don't really know all the potential channels, it seems like it's very difficult to think that we can actually do something to...
Well, I think in the case of a trait like longevity, what happens if you control for, I mean, you have to model, longevity is a particular trait because you have to be careful to model it properly. So I agree, you could probably just control for age and, I mean, if you don't, I mean, you'd have to do it in the proper way. But for a trait, let's say, think of income and height, again, this example. Well then, we know that genetic variants don't correlate with age. I mean, there might be some variation across cohorts, but it's very weak. You know, it's not as though we're changing dramatically. Genetic variants change dramatically from cohort to cohort. So partialing out age, all it will do is remove part of the variance from the noise, essentially. If that makes sense.
Well, I was thinking that in a sense, as you age, you become higher, but if the probability of you surviving is also affected, and both of them you may be like biasing the sample in a sense, and that's where I was thinking it may be different.
I see. Yeah, I mean, I guess that goes to a more complicated question about sample selection bias, but I mean, there might be a case where you can imagine that like it's not a good idea. I think in general, age and sex are orthogonal to the genetic variance, so it's good to partial them out. But we can talk more after. Yep.
So we really talk a lot about how we control for population stratification, for LD, stuff like this. Why do we seem to always like gloss over the parental environment thing? Like, is there a reason for that?
You mean in what's? Can you be more precise? What would you?
How is that controlled for? And if it's not, why do we ignore it?
You mean you would like to control? You're asking why we don't control for parental environment in let's say this regression. Well, it depends a bit on what we want to get at. But here, what we want, let's take this example of height on income, okay? What you want to you want to be able to induce exogenous variation in height. So you want genetic variants that change height, but don't change income directly, basically, right? And if you have that, then just regressing income on this instrumented height should give you a causal effect. Then there's just no need to control for parental environment in this setting for the question that we're interested in. And there might be other questions where it's worthwhile to control for parental environment. But does that address your question? Okay, we can talk more later.
But in this same example, if my parents' income affects my income, let's say via education, then you really need to control for the parents, right? Because again, the genetic endowment is affecting their height, which may be affecting their income, which is affecting my income.
Oh yeah,
right?
yeah. That was my example here that I skipped. Yeah, basically this example that I skipped. So in this case, actually, I think controlling for this would still not solve the issue. Well, if you knew exactly what you control for, maybe. But basically here we just have a failure of the exclusion restriction. Yeah. OK, so now let's cover a few examples. And as I said, I'll be calling on you to tell me what you think of these. And the answers are not in the slides. So here's a paper by Tyrrell et al. In BMJ 2016. It's about height, body mass index, and socioeconomic status, Mendelian Randomization, the UK Biobank. So they analyzed 120,000 individuals of British ancestry aged between 37 to 73 years old in the UK Biobank. And the exposure variables are height and BMI, okay? And they analyze these separately, okay? And the instruments are genetic variants. And for height, they use essentially a polygenic score constructed using 396 independent variants that had been identified by Wood et al. In the largest to date GWAS of height. It was the largest when they published this, and I think it's still the largest, right? For BMI, they created a polygenic score constructed using 69 independent variants identified by the largest GWAS of BMI to date by Locke et al. With 339,000 individuals. The outcome variables are two measures of educational attainment, which are age completed education and degree level. So age completed education, you probably haven't, many of you might not have encountered this. Essentially, it's highly correlated to number of years of education. Okay, so when I say age completed education, you can think of educational attainment in terms of number of years of education. It's highly correlated. And then they also look at job class, household income, and Townsend deprivation index. The latter is an index of social deprivation, socioeconomic deprivation, such as, actually I'm not the expert on it, so I won't talk on the record on. But anyways, it's an index of social deprivation. So the higher you are, the more depraved you are socioeconomically. Okay, so essentially what they're asking in this paper is what's the causal effect of height on those socioeconomic variables? And what's the causal effect of BMI on those socioeconomic variables? So if we could randomly allocate height to people in this class and to a larger sample, and then follow you up for the next 40 years and see how your income varies based on that, that would be basically the experiments we'd like to get. We'd like to basically vary height exogenously and see how this affects income, okay? And the way to do this in practice is they use a polygenic score of height, okay? And then for BMI, they do the same thing, okay? They want to see what's the causal effect of height of BMI on those socioeconomic status. Okay, here are the results for height. So this table is for height, taller stature, and after we'll see the table for BMI. Okay, so in this table, first you have, the first column here shows the 5 different outcome variables they analyzed, and for each of them, they analyzed them for the whole sample, for men only and for women only. And first they report the observational effect, so that's just regressing the measure here, age completed, on height, okay? And then they look at the genetic estimate based on Mendelian randomization. Okay, so let's focus on this quadrant first, on this set of results for age completed education. And again, I repeat, this is essentially a measure of educational attainment, highly correlated with educational attainment. Yeah?
So if I'm thinking of something like the polygenic score, right? And I'm looking at the population as a whole, should I think of that as randomly assigned across the population? I thought that I should think of it as randomly assigned, conditional on the parents' genetic material. Is that not right, or is that randomly? Should I think of that as randomly assigned across the whole population?
No, that's a good question. So I guess well, here we are thinking of it as randomly assigned across the population. So basically, we're assuming there's no population stratification. So population stratification would be when there's correlation between the polygenic score and cultural variables that affect the outcome of interest, right?
So I think one issue, I think ideally you would do Mendelian randomization within families for exactly the reason you're
Yeah.
saying. And the problem has been a sample size limitation. And so the way these studies have proceeded is by controlling for principal components and then acting as if, you know, the genetic variants are now randomly assigned conditional as principal components, which is kind of the same thing we do in GWAS for the same reason.
Absolutely, absolutely. No, the ideal design would have family fixed effects. As Dan said, it's a sample issue. Like the data sets that have family data are rare and small. So basically, if you want to use these to do this analysis, your power will be severely restricted. Actually, we'll see an example later that uses family fixed effects. Okay, so here, what we do see is that observationally, an increase, so these are all in standardized coefficients. So basically, this tells us that a 1 standard deviation increase in height increases age-completed education by 0.11 standard deviations, okay? And so basically, there's a positive correlation between height and education, okay? And that's for male, for the whole sample, for male and for female. However, when we use Mendelian randomization, this effect decreases a lot, and now becomes 0.03 for the whole sample, which is equivalent to about a month. So 1 standard deviation in height is, based on this, seen to causally increase education by about a month, okay? And that's significant for the whole sample. It's also very significant for males, more so than for female. And for female, it's not significant at all, actually, okay? Now, job class, these are odds ratio, but we see the same picture, basically. Observationally, there's an effect that's positive, such that height increases status. And then when you do Mendelian randomization, it's significant for the whole sample and for males, but not, actually, in this case, it's significant for females also. And then the annual household income, we see again the same picture that there's the observational estimate suggests that height increases status. And then when using Mendelian randomization, the effect is smaller, but still non-trivial and significant for the whole sample, for males, but not for females, or at least marginally so for females. So based on this, if you believe that the exogeneity condition is satisfied, well, we find that height causally increases socioeconomic status, okay? Now, actually, that's when I'd like to ask the class, what do you think about it? Do you think, would you believe these estimates? Does height causally increase socioeconomic status?
Is there a problem with highly polygenic traits and pleiotropy in this case?
Yeah, yeah. So basically, that would be the exogeneity condition failing, based on what you suggest, because there may be pleiotropy. So what if these 396 markers for height, some of these also impact, let's say, cognitive performance directly, you know, or whatever you want, whatever other trait you want that affects income. Well, then these estimates would be biased, okay? So the difficulty here is we just don't know whether this assumption is really satisfied. And it's very difficult to be confident of it, short of having much more advanced biological knowledge, and even then, yeah.
Could we then just control for the polygenic score for educational attainment and then pick some of the problem?
Yeah, no, that's a good idea. So the difficulty is the polygenic score for educational attainment is very imperfect, right? It doesn't explain the full genetic variation in educational attainment. Now, however, you're hinting at something. Actually, there's a colleague of Casper in the back. Where is he? Yeah, has been working on a similar method that's basically the idea of the method, is to control for the polygenic score of the outcome variable in this way.
So, I mean, one of the issues, though, if you start going down that path of controlling for the polygenic scores for different pathways, is we're kind of back in the world of an observational study where you're trying to plug the holes by including additional control variables, and we're no longer in this, you know, we no longer have this beautiful setup where we can think of kind of getting the causal effect because we have some random assignment.
Yep.
But you could basically use the plausible exogenous type of test. I mean, it seems like there are a lot of, I don't know if this is the only thing they show, but I mean, first of all, I would like to see some reduced form regressions to see.
These are the reduced form. This.
No. Those are the OLS, right?
Yeah.
Not the reduced form. Reduced form, I mean, output directly on the genes. Not
Oh, I see.
Right? So just to get also a sense of weak, and I guess they didn't show us F-stats or anything, so it's hard to tell. But to overcome the previous problem, one could try to look at plausible exogenous and then see how much of a violation of exogeneity you would need to basically get rid of the results, right?
Yeah. Yeah, actually, a method that kind of does that is Mendelian MRG regression. I mean, it doesn't do exactly that, but we'll cover that very soon. It kind of gets at that idea, I think. So yeah, so anyways, the issue with this is that it's difficult to know that the exogeneity condition holds because there might be pleiotropy or a linkage disequilibrium or, again, parental effect or, on the other hand, at least to give them credit, to give them proper credit, the relevance condition here almost certainly holds because the genes here that they use, or the genetic variants, are the 396 markers from a large-scale GWAS of height that were genome-wide significant. So I'm not worried about the relevance condition, at least. There was a question in the back? No? Okay.
So Jonathan, just along these same lines people have been discussing, I mean, first of all, genetic correlation in height and IQ, for example, is not very high. It's like 0.2 or something like that. But I mean, I'm just thinking probably of those 390 variants, none of them are genome-wide significantly related to IQ. But I mean, could you take just a subsample of those that are the least related to IQ and rerun it and see if you're getting basically the same results? And if you are, wouldn't that suggest that this issue of pleiotropy probably isn't biasing our results very much? So you take that.
Yeah.
You know,
Yeah, no, I agree. I mean, if you basically look at these 396 and see which one of these you have good biological knowledge and, you know, you look.
Aside from biological knowledge, just, well, maybe we're talking the same thing, but just statistical evidence that there's no relationship between those and IQ.
You'd have to have a very large sample to be sure of that, but yeah. But
Well, I mean, with a sample, I mean, in some of these huge meta-analyses that are being done on IQ, I think you're getting a fairly good
Yeah, that's
understanding
true.
of the
Yeah.
ones that are probably, you know, if they're related, they're related.
But they could
very
impact other
very
things
close.
such as self-control, time preferences,
Yeah.
you know, personality trait, anything
Okay,
that.
right.
So
Yeah,
that's the
yeah,
difficulty
yeah,
is
right,
you'd have to rule
right.
out all these effects.
Right,
one by
okay,
one.
I see what you're saying.
Yeah. Okay. Okay. You and then do this.
You were saying that you don't worry about the significance given that you have GWAS results, but given that the effects are small, that they are squared are small, in the end, it could still be that these are really weak instruments,
Yeah.
right?
Yeah.
So
So actually, good point. So I don't worry about the relevance condition because these are from the GWAS, but also the R squared of those 396 variants. I forget what they are. James or someone, do you know? It's like it's like 10% or something. It's non-trivial.
yeah, but still, let's say if you control for certain type of fixed effects or other things that you think are important to control for, the additional R square, like the instrument may become weak even though in the first, you would think that in the first stage it is.
Yeah, they should do an F test. I don't think they've done it. I'm not too worried about it because they control only for age and sex, and like basically this incremental R squared of these variants of 10% is after partialing these out. But
And they have 120,000.
yeah, and it's a very large sample size. Yeah. Okay, I'll take one more question, then I'll shoot ahead a bit because I'm standing between you and lunch. I don't want to. Yep.
I'm just interested in understanding what sort of the consensus or view is in the field, because this, to me, it seems like it's a bit of a dangerous path in the sense that it's not clear that you can basically know that this works, no? So what's the idea? And
I think it's debated. I think if you ask different people, you'll get different answers. So I think the consensus is evolving. Anyone, Dan, do you want to comment on that?
My sense is that there's that geneticists typically are very excited about this because it's like a potential usefulness of the GWAS results that can start answering all sorts of causal questions that seem like they weren't possible
yeah,
to
at first
answer.
it's exciting.
Yeah, and I think that they understand the problems that Jonathan is talking about, and there's now a lot of efforts to develop various elaborations of the method that get around one or another of the problems. I think Jonathan's going to talk about MRAGR, which is one of those. And I think, some of us in social science are on the more skeptical end because we know that, that
We've been through this.
We've been through this, and we've tried to, plug the holes, and we think, ultimately you just really need, in the end of the day, to have really good, in this case, biological knowledge to be able to make a strong case for the exclusion restriction, and there's no kind of statistical way around that. But I think there's still tension.
So in the future, when we actually have more biological knowledge, we might be more confident? Because that's interesting.
I mean, I think that this is very promising in cases like
We'll see one soon, yeah, nicotine.
example Jonathan's going to talk about soon with the nicotine receptor SNP. where, you know, we know what it does biologically, and it seems plausible that the effect of that SNP on smoking is just so much bigger than anything else that SNP could potentially be doing or be correlated with.
Yeah.
But you know, that's not statistics. That's really understanding the mechanism and making a case for the exclusion restriction holding.
So is there a statistical way to like prove the exclusion restriction, or you just kind of have to let story?
There are some tests for over-identifying restriction, but they're not. It's not the same thing. Basically, if you have if you're over-identified, basically if you have more instruments than instrumented variable, you can test that these are consistent with one another. And I mean, so there are some tests, but they're not. I mean, they always assume that at least one instrument is valid or something like that. Yeah. So yes, ultimately you have to make a case, a non-statistical case for the validity. I want to keep going because, okay, so then for BMI, this is the same table as before except now for BMI. Okay, it's written in small here in case you're curious. I won't go through the details, but the main point I want you to is just summarizing the results. What they find is observationally, BMI is negatively associated. Let's focus on household income here. BMI is negatively associated with household income. And then, however, not for males, or at least not very much for males, mostly for females. And then when they do MR-EGR, they also find that the effect is much larger for female, actually. So basically, stouter female seems to be penalized in the labor market, okay, based on this. Actually, this is household income, so it could be that they have a marriage partner that makes less money also. I mean, it's unclear that it's themselves. But then if you look at Townsend Deprivation Index, again, there's this negative effect. So based on this, if you assume that the relevance and exogeneity condition hold, then there's a negative causal effect of BMI on socioeconomic status. Of course, as I said, this is assuming those assumptions hold. Okay? Okay, here's another example. This is entitled Genetic Lotteries Within Family by Fletcher and Steve Lehrer in Journal of Health Economics 2011. They examined the effect of poor health on academic outcomes using the HAD-HEALTH data set. Now here, poor health is depression, being overweight, and ADHD. It's a within family design, so basically they use family fixed effects, okay? Which ensures that population stratification does not drive the results. And they use genetic markers to instrument poor health. So they use 6 markers that are 6 candidate genes, one for the DAT, dopamine transporter gene, one for dopamine D4 receptor, DRD4, one for serotonin transporter 5-HTT that we saw yesterday when Matt was talking about Caspi et al. And then one from the monoamine oxidase AMAOA and dopamine D2, DRD2, and this one here. So these are some of the most commonly used candidate genes. So here, basically what they want to see is whether poor health, which is these 2 traits, depression, overweight, and ADHD, has a causal effect on academic outcome, okay? And they use these genetic markers to instrument poor health, okay? The problem here is that this marker may well impact poor health, but they may also directly impact academic performance. Like for instance, the dopamine plays a role in learning, so I wouldn't use these as in this setting. Also, the relevance condition might even fail here because these are candidate genes. So as we know, actually I don't know the evidence for these on health now that we have larger samples, but in any case. To their credit, Fletcher and Lehrer recognize this. They're good economists, and they write in the paper that these caveats, okay? But yeah, there's a question?
Yeah, I mean, so I was going to raise this kind of issue, but I still just wonder, like, what exactly justification they could bring, even acknowledging it's imperfect, but like, I mean, DRD4, DA21 are suggested to be, and DRD2 are suggested to be associated with ADHD, okay, not necessarily the overweight or depression, who will be comorbid, and it's just like a really strange thing to like pick these 6 and like.
Yeah, so I think data limitation was an issue here. They used the HAD-HEALTH, and so they have a very small sample, and they had to. But yeah, I think they would agree with you if you talk to them. Yeah.
Were they just trying to get like some number, like so that maybe just using a bunch of different variants? Obviously, we're trying to hear about more like using 500,000, but what's the idea that like using 6 would be better than just using 1 or 2?
Well, I mean, this paper was published in 2011. In economics, that means the paper was probably written in 2007 or 8. So back then, you know, that was reasonable. So, I think to be fair to them, they tried to pioneer economic technique and genoeconomic data back in the very early days. You know.
I think it's important to point out that first of all, this was one of the very first Mendelian randomization papers in social science. And also, this was before it was widely understood within social science that the candidate genes were problematic. Remember that editorial policy from behavior genetics that I showed you last week was from 2012. And as Jonathan says, this paper was actually written a few years before 2011.
Yeah, yeah. Okay, finally, an example where I think this is plausible. So does high tobacco consumption cause psychological distress? That's a paper by Skov, Etrop et al. In Nicotine and Tobacco Research 2017. They analyzed 90,000 individuals in a Copenhagen population study. The exposure variable is self-reported cigarettes per day that you smoke, and pack years. Pack years, basically smoking 1 pack of cigarette every day for a year, that counts for 1 pack year. If you do that for 2 years, that counts for 2 pack years, and so on, okay? So basically, the exposure variable is smoking, okay, amount of smoking. The instruments are SNP RS1051730, which tag the CHRNA3 gene. And that variant is known as Mr. Big because of its relatively large association with the amount of smoking and nicotine dependence in current smoker. So in US smoking, it comes out as Mr. Big, it really pops out. It causes an amino acid change in the nicotinic receptor, which alters the responsiveness of the nicotinic receptors to nicotine. So not only is this variant known to have huge effect on smoking and nicotine dependence, but the actual biological mechanism is understood, okay? So I think that's as good as it gets. The outcome variable here is psychological distress, stress, fatigue, and hopelessness. And what they want to know is whether cigarette smoking has a causal effect on psychological distress. And they use this Mr. Big variant to instrument smoking, okay? So I think this is one design where it's really well done, and well, it's really well done, where it's really convinced. Well, I'm not an expert in the genetics of smoking, so I don't want to emphasize this too much, but a priori for me, it sounds more plausible than many other cases, okay? Because you have a variant that has a large effect on smoking and whose biological mechanism seems to be understood, as far as I know at least. And unfortunately, they find nothing. Failed to find an association between psychological distress and RS105. So basically, there's no effect. Okay. But I think for those who are curious, interested in Mendelian randomization and are interested in smoking, studying Mr. Big is a promising avenue. Now, I think if everyone starts doing study like this, something significant will come out. So make sure you have large enough data sets and you pre-register. But okay, good. Let's talk. Yep.
So if they didn't find any effect with this, like psychological distress, did they try doing it on other outcomes and see if?
Not reported in the paper as far as I remember.
It's a really big effect if you do it with lung disease. So you can what you'd expect to see.
Proof of principle.
Yeah. So that's large enough that you can see it in the HRS.
Oh wow. Well. So yeah, Mendelian randomization can work, I guess, very well for. I mean, proof of concept, as you said. Yeah. Yep.
In your example 2, right? Let's say if there is you found a polygenic score for poor health or a particular disease condition, could you then use that as
if you find an instrumental variable, if you find a marker for poor health, well, you could if you are confident that the exogeneity condition holds, basically. So basically you have to make sure, it goes back to what we were discussing earlier, you have to make sure this marker impacts poor health, but does not directly impact or is not directly associated with academic achievement aside from via poor health. Yep.
Which variation does Mr. Big explain this?
Good question. Anyone knows what's the R squared of Mr. Big?
3.4.
What?
0.4. Yeah.
0.4%? Yeah, well, almost 0.5%, yeah. Okay, good. So let me now talk about MR-Egger regression. So MR-Egger regression is a technique developed and popularized by a paper by Bowden et al. In the International Journal of Epidemiology 2015. It's an alternative method to estimate the causal effect of the exposure for Mendelian randomization studies. So it's another way to get at beta based on Mendelian randomization. Dan was saying that, like, in response to the question as to what's the status, this question about what's the status now in the field, what do people think about Mendelian randomization? Well, Dan said people are aware of these issues and there's lots of efforts to find better methods. Well, that's an example of such a method. I think probably the most promising, the most popular one. The name comes from the fact that this regression is similar to an Egger regression. That's a regression that's used in meta-analysis. It's just for your information. So MR-Egger is Mendelian randomization Egger regression. Okay, that's just the name anyways. Okay, so now the framework. There's lots of stuff on this slide, so let's go through it in detail, and then I'll use those old graphs again to clarify. So the first equation is y is equal E beta plus epsilon, and that's the same as before, okay? That's equation from before when I introduced Mendelian randomization instrumental variables. The second equation is E is equal xj gamma j plus xi j. That's the same as before, except now we have subscript j. So basically, we'll have one equation like this for each variant j, okay? The third equation is just plugging the second one into the first one. So what we get is y is equal xj gamma j plus xi j beta. So that's just from plugging this into this. Plus epsilon, which is equal to xj gamma j beta plus the error terms, which we'll call ej epsilon j tilde, okay? And the other thing we do is we'll align all the xj's so that gamma j is greater than 0 for all j's. Okay, that's without loss of generality. We're allowed to do this. We'll just code the xj's in such a way that they're all gamma j is positive for all of them. Okay, so now if one regresses if one regresses y on xj, the estimated coefficient on xj will be the following. Will be gamma j hat, which is equal to the empirical covariance of xj and y over the empirical variance of xj. Okay, this here is basically just the definition of regression coefficients. And let me write this down just to make sure this is clear. I think the green pen doesn't seem to work too well, so I'll use hopefully the purple one goes better. So basically what we do is we regress y is equal, what did I say? Yeah, is equal rj xj plus, let's call it uj. Okay, so we estimate this here. And then what I'm telling you is that gamma j hat is equal to the empirical covariance between xj and y over the variance of xj, the empirical variance of xj. So that's just the definition of OLS coefficient, okay? Now, if we have a large enough sample, this will converge to gamma j beta plus the covariance of xj epsilon over the variance of xj. Again, this is just from using OLS. If I were to substitute in the covariance, if I were to substitute in this expression, if I were to substitute y here in this expression and just work out the details, that's what I get. And you'll do that in problem set, I believe. Okay? So basically, this is equal to gamma j beta plus alpha j, okay? So here, alpha j captures the direct effect of xj on y, independent of E, okay? And we'll see that graphically in the next slide. It can be thought of as a measure of pleiotropy for xj, although LD, parental effect of population stratification, could also be involved, okay? Okay, so just to recap now, we'll go through this in more detail next slide. What I want you to get here is that we have these three equations, and then we regress y on xj, and we get gamma j to be equal to this here just from basic property of OLS regression and plugging stuff in. Okay, graphically, here's what's happening. We have, again, I'm just repeating those equations. We have y is equal E beta plus epsilon, E is equal xj gamma j plus xi j, and gamma j hat is equal to the estimated coefficient of xj from regression of y on xj, okay? So basically, gamma j captures the direct effect, the total effect of xj on y, okay? And can be written as being gamma j beta plus the covariance between xj epsilon and var xj, okay? here's the way to see what this is, basically with colors. The first component here is gamma j beta, is this here. Okay, so that's the effect of xj via E. Okay? And then alpha j is the effect of xj on y that's not via E. Okay? So it's the covariance between xj and epsilon. Okay? So basically, this coefficient gamma j hat is equal to the direct, the effect of xj via the exposure, and then the direct effect not via the exposure. Okay? Everyone sees that? Okay. Now, remember what the exogeneity condition says? What would be what would be alpha j if the exogeneity condition holds? 0. Yeah. The exogeneity condition says that x is orthogonal to epsilon, so under the exogeneity condition, alpha j is equal to 0. Okay? Now, the beauty of MR-Egger is that it doesn't require the exogeneity condition. It works, it doesn't impose that alpha j is equal to 0. Instead, what it will impose is that the correlation or the covariance between alpha j and gamma j is equal to 0. So what it imposes instead, what it assumes instead is that there's no systematic relationship between gamma j and alpha j. And as such, alpha j will not systematically bias the estimate of gamma j. Okay, we'll go through this in more detail, but that's the overall intuition, okay? So it basically is more general. Instead of just assuming that alpha j is equal to 0, it says we can still live with alpha j not being equal to 0 as long as it's not systematically related to gamma j. Actually, I want to, yeah, okay. So now the inside assumption is the assumption that basically on which MR-Egger regression is based, says that the covariance between gamma j and alpha j is equal to 0. So the effect, what it says essentially is that the effect of the xj's on E, or the gamma j's, are uncorrelated with the direct effect of the xj's on Y, which are the alpha j's. Okay? And insight stands for instrument strength, which is the gamma j, independent of direct effect, which are the alpha j. Okay? Okay, again, this little graph. Okay, that's actually, I already went through this in the previous slide. Basically, inside says that gamma j is independent of the alpha j. So the instrument strength, the gamma j, okay, is independent of the direct effect, okay? And as I said, inside is weaker than the exogeneity condition, okay, because the exogeneity condition implies that alpha j is equal to 0, which in turn imply inside. So basically, when we have exogeneity, we have inside. But when we have inside, we don't necessarily have exogeneity. So another way, inside is more general, and so MR-Egger is more general. It's more flexible, so to speak, okay? Now, I'd like to illustrate this briefly, say, to think when the inside assumption holds versus not. So let's think of our example from earlier of, suppose we want to know the causal effect of height on income, okay? So here, E is height, and Y is income, okay? And these are genetic variants that affect height. Well, the question is whether, we saw earlier that there's many reasons to think the exogeneity condition would not hold in this case because the variance for height may be pleiotropic and may impact labor market skills directly, right? Now, do you think the inside assumption would hold in this case? Anyone wants to take a try at this? It's not on the slides. Anyone? Bonus points? Okay. So I'll give it a try. So, I mean, we don't know, again. So if you think that, okay, think of a simple model where there are variants for height that affect bond length, and they also, there's pleiotropy, and they affect, let's say, cognitive performance as well. Okay, so cognitive performance would be this indirect effect here. The effect of the variance on cognitive performance would be this indirect effect. Now, if you're in a world where the variance effect on bone length are independent of the variance effect on cognitive performance, then inside holds, okay? So basically, if the variant makes you taller, it also has an effect on cognitive performance, but could be negative or positive. You know, there's no systematic relationship, then inside holds. However, if we're in a world with a variant that makes you taller, also typically makes you cognitively have a higher cognitive performance, then alpha j will be correlated to gamma j, and inside will fail. Okay? Yep.
Just wondering, like, so
with sort of correlation and pleiotropy, I guess I guess we're also talking about decrease of pleiotropy as opposed to whether there's actually pleiotropy or no pleiotropy, because.
You said decrease of pleiotropy?
Yeah, I mean, I'm just. So if, there was a paper recently which was mentioned over here which suggested that, there's an omnigenic model,
Yeah.
Extreme sort of pleiotropy, extreme sort of polygenicity and things like that. So in this case, if we have like effects on both the traits for a set of SNPs, but the effects of the set of SNP on trait P is actually really, really small, would that really matter?
Well, if it's really small, I mean, I think if it's negligible, I mean, it's all relative to the estimate you care about. So I mean, there are cases where the pleiotropy, there might be pleiotropy, but like it's so small that the bias is small. But I mean, it really depends on and how it works out for the final estimate. Yeah.
Now here you're taking the x's to be like the SNPs, right?
Yeah, so yeah, you can think of them as SNPs.
Okay, so if it were the SNPs, then let's say that imagine that a whole bunch don't have an effect on one of the two traits that you're worried about, right? The indirect. Then you will start having a correlation just because it won't have an effect on most. For most, you won't have a correlation but, on effect, but then for some, yes, and then suddenly you will have a correlation, right? Which would create a violation. Now, I guess for practical uses, you would actually use the one of the GWAS constructed polynomial scores, right? But for the polynomial scores, we actually don't have an idea. I mean, a lot of people are just trying to focus and construct whether the polygenic score affects certain things, right? And from there, constructing the polygenic score. But we don't know if those polygenic scores now correlate with a whole bunch of other things, right? Which again may violate this.
Yeah. So in practice, what happens, the way we do MR regression typically is we take, let's say, genome-wide significant SNP, let's say from the GWAS of height. Think back of the example in Tyrrell et al. There's 396 of them. So for each of them, actually, for each of them, it's a marker. And so basically the question is, for these, do they have pleiotropic effect? But you're right, if we were to use this with all the markers that we include in the score, when you use all markers, then that would be almost hopeless, because there's for sure there's going to be pleiotropy then. Okay, good. So now, what's the MR Egger regression now? So bear with me, we still have for about 5 minutes, I think, so before lunch. So the MR so finally we get to the MR regression, which is what we ultimately care about. So recall the inside assumption states that covariance between gamma j and alpha j is equal to 0. Now we have the data from gamma j hat and, well, actually, sorry. Yeah, this is, I guess, capital gamma j hat. Yeah, it's confusing notation, at least when you talk about them verbally. This is small gamma j hat and capital gamma j hat. Okay, and you have these for all the markers from 1 to j, for all the markers you think are valid here, from 1 to j. Well, and remember, capital gamma j is from this regression, and small gamma j is from the regression of E equal x, or rather, gamma j xj plus xij. Okay? So basically, the capital gamma j is the reduced form estimate of xj on y, and the small gamma j is the effect of xj on the exposure. Okay? So what we'll do essentially is we regress capital gamma j estimates on small gamma j, okay? And then beta E hat is going to give us an estimate of beta. And here's the math in between, which I think you go through in the problem set, if I remember well. So basically what happens is that, I'm just reading through this, beta hat sub E is equal to the empirical covariance between gamma j hat and capital gamma j hat over the empirical variance, the sample variance rather, of gamma j hat. That's just from OLS properties, okay, that's the definition of OLS coefficient. Now, if we were to have very many SNPs or variants, well, this sample covariance becomes the true covariance, okay, so we get rid of those hats. And then if the number of individuals in the GWAS gets to infinity, then these hats here become the true estimates, so we get rid of those hats. And then working this through, we get that this is equal to beta plus the covariance of gamma j, alpha j over the variance of gamma j. But here, this is where the inside assumption comes in. The inside assumption says that this is equal to 0, okay? So this is equal to beta, Okay? So in other words, MR-Egger regression gives us another way to get an estimate of beta. Okay, so beta here is an alternative to the IV estimator. It is consistent under weaker assumptions, consistent under the inside assumption instead of just the exogeneity condition. Okay, I won't cover this in class, but you can go through it in detail on your own. The point is MR-Egger regression gives a test. The intercept of the MR-Egger regression, when it's not equal to 0, tells us that there's what they call in the paper directional pleiotropy. Okay? Okay, so here's an assessment of this. So the advantages of this regression is that if the inside assumption holds, it yields consistent estimate of beta. By contrast, the two-stage least square, I should say IV here, not two-stage least square. It's the same thing, well, generally speaking. So the IV estimates are biased when the exogeneity condition fails, even if the inside assumption holds. If inside holds, MR-Egger's test can detect failure of the exogeneity condition if well-powered. That's the slide I just skipped. Okay? However, there are some limitations. So first, it relies on the inside assumption, although that's better than relying on the exogeneity condition. However, it has also reduced statistical power relative to IV, okay? Because now you just have a limited number of SNPs in the regression. Also, how to determine standard errors. Okay, so back to the equation here. What they do in the paper is they just report the standard errors as the standard errors from this regression. But this regression uses noisy terms, right? So those standard errors will be downward biased. So, I mean, there are adjustments to that, but one would have to think a bit more about how to do this properly. Also, another limitation is that this assumes independent markers. If you don't have independent markers, it's a bit complicated because, well, we can discuss about this in more detail later, but basically this assumes independent markers, okay? So basically, it's quite an interesting method that can work if the inside assumption holds, but there's still some limitations even there. Okay, here's an example from the literature. Actually, this paper was published in 2013, before the MR-Egger regression, although it kind of uses that, even though it doesn't call it this way. So in this paper, it's by Doe et al., Nature Genetics 2013, and they care to see the causal effect of cholesterol, HDL or LDL, and triglyceride on cardiovascular disease. So basically, they want to see the causal effect of cholesterol on cardiovascular diseases, okay? And what they did, essentially, is they studied 185 SNPs with p-value less than 1 × 10⁻⁸ for triglyceride, LDL cholesterol, and HDL cholesterol in a meta-analysis of 188,000 individuals. And let's focus, what have I just done? I think I pressed the wrong button here.
That wasn't me.
No, that's me. Okay, so let's focus on the first set of estimate here, just the first square. So what they did here, this is beta CAD. So that's essentially corresponding to this guy here, the gamma J. So that's the estimate of regressing cardiovascular disease on the variant. Okay, so that's the reduced form estimate. Now this is beta LDL cholesterol. So that's the estimate for regressing LDL cholesterol on the variant. And here, this is the beta from regressing this estimate on this estimate. So essentially, this is the beta from MR-Egger regression. And what they find is that there's a significant positive effect of HDL cholesterol, here it's LDL cholesterol on cardiovascular disease with a very, very small p-value. I mean, this is a very small p-value given the number of variants of observations. Okay? So this is an example that used MR-Egger regression before MR-Egger regression. So, and they find, this seems plausible as far as I know in the literature. I think that cholesterol causes cardiovascular disease. I mean, I'm not the expert again on this, but that might be a plausible, it might be a plausible case, okay? I'd have to think and to read more about it. Okay, to conclude.
Sorry, John.
Yeah.
So one thing that Patrick has pointed out as a potential major problem with MR-Egger regression is
the LD thing, yeah.
the LD.
Yeah, okay, yeah, no, good. I'll mention it then. So actually, Patrick mentioned a case where the inside assumption can fail. Let me get to the right. Right, okay, we're back to this inside assumption. Okay, so suppose that the SNP's effect on trait 1 is proportional to its LD score, okay, which makes sense. Its US coefficient is proportional to its LD score. And its effect on trait 2 is also proportional to its LD score, simply because when it has a high LD score, it captures more causal SNPs, right? Well, then you will have pleiotropy, or well, it's not really pleiotropy here, it's kind of LD. But basically what it can create is that the direct effect of the SNP will be correlated with could be correlated with the indirect effect, okay? So this is a condition where it fails. So under that scenario, you'd have to have the effects to be aligned, basically.
Yeah. I mean, if you're willing to admit that there's directional pleiotropy, which MR does, so the whole reason that you put in this intercept is because you think there may be some directional pleiotropy, and the moment that you think there may be directional pleiotropy, what kills you is when the direct effect or the pleiotropic effect is proportional to the to the first stage effect, the effect on treatment, and because that's going to be that's what we mean when we say they're correlated. So when one's big, the other one's big. And when we were talking about LD score regression, what we're saying is that, well, if a SNP tags a lot of other SNPs, it's going to tag a lot more causal variation. So this is exactly the problem. Because we're tagging more causal SNPs potentially, then when we see large first stage effects, we're also going to see larger pleiotropic effects. And so that's a concern that just in general, if you do MR I don't think that it could hold generally.
I mean, I think you really would need to do this something like within LD score bins or something to somehow control for LD, which I don't think that people have explored yet.
Yep. I think, however, you'd have to assume some directionality also to the two traits, like in the sense that those that are if the effects of trait 1 and trait 2 tend to be in opposite direction across the genome, then that thing would not. I think the inside assumption would still hold, right? Because then the SNP that have higher LD would have larger magnitude effect on trait 1 and trait 2, but the direction could be.
I mean, let's talk about it maybe.
Yeah, okay, good. Yeah.
Would it still lead to a bias? It's just the opposite bias? It gives you an underestimate of the effect?
No, I think actually, let's talk about it after, I think it will be. Okay, good. So concluding remarks. So Mendelian randomization could in principle help researchers identify the causal effects of exposure variables that are typically associated with confounders. However, it relies on strong assumptions. Most importantly, conditional on the exposure, the instruments must be independent of the outcomes, which is the exogeneity condition. So there are many situations in which this may fail, including population stratification, pleiotropy, LD and parental influence. The MR regression yields consistent estimates as long as the inside assumption holds, even if the exogeneity condition fails. And I think, it also has limitations, however. It requires the inside assumption. It has reduced statistical power. Standard errors only hold asymptotically. But still. So thank you.